So today we'll be solving question 5 of May 2023. Stents are used in the treatment of atherosclerosis. Stents widen the diseased coronary artery so that blood can flow through to the heart muscle. The diagram shows a stent, diseased coronary artery and the stent. Stents can damage the endothelium cells lining the artery and trigger the formation of a blood clot. Patients who have a stent fitted are given anticoagulants. Explain how damage to the endothelial cells result in the formation of thrombin. So how thrombin is formed? Thrombin is formed from the thromboplastin, right? Thromboplastin result in the formation of thrombin from prothrombin, isn't it? So how thrombin is formed? It's basically because the platelets contain or it releases thromboplastin, which result in the formation of thrombin. Okay. Platelets releases thromboplastin, which converts the thrombin into thrombin. Give one risk of treating patients with anticoagulant. If we give a patient anticoagulant, then they can bleed too excessively or internally. Okay. The person bleeds excessively. Okay. Question number 3. C. The photograph shows a tropical bond ring. This bond ring produces a peptide called verigin. It produces a peptide called verigin. Verigin binds to the active site of thrombin and prevents blood clotting. Verigin is a peptide consisting of 32 amino acids. It consists of 32 amino acids, okay? Which is the minimum number of bases in the gene that codes for this peptide bond. So it contains 32 amino acids, right? So three bases are required for one amino acid. So the answer will be D, 99, because the three codons are required for one amino acid, right? Three codons are required for one amino acid. So 32 will be requiring for 3 2 is 6, 3 3 is 9, 96 plus 3 stop codon. So total 99. I hope I made it clear. So it says explain how verigin prevent blood clotting. So how verigin works? Verigin binds to the active site of thrombin and prevent blood clotting. So if verigin binds to the active site of thrombin, it will not be able to convert fibrinogen into fibrin. Correct. It will not be able to convert fibrinogen into fibrin. Correct. So, and without fibrinogen, the platelets or the blood cells will not get trapped. Okay. As verigin binds to the active site of thrombin, hence it cannot convert fibrinogen into fibrin, and without fibrin, platelets, blood cell will not get trapped, forming a blood clot. Okay. Thrombin slowly splits verigin into two molecules. Explain how thrombin splits verigin. Okay, so as verigin binds to the active site of thrombin, so it makes an enzyme substrate complex, right? So thrombin lowers the activation energy, and therefore peptide bonds are broken by thrombin by the hydrolysis reaction, and by that a variegin gets split. Okay, variegin binds to the active site of thrombin, and thus an enzyme substrate complex is formed. Thrombin lowers the activation energy, therefore the peptide bonds are broken by thrombin by hydrolysis reaction. Okay, for the effect of variegin on the mass of blood clots forming inside the stent was investigated. The masses were compared with another anticoagulant drug H. The graph shows the result of this investigation. Each plotted point represents the mass of blood clot in one stent. Mass of blood clot. Okay. Variegin and drug H. Comment on the result of this investigation. So you just need to make a comment of the result. Whatever you see in this diagram is that, like, overall variegin result in less blood clot than drug H, right? Then there is less variation in the size of blood clot in patients treated with variegin. Okay, so let's see the answer. There is less variation in the size of the blood clot in patients treated with variegin. Cannot comment on the effectiveness of the variegin compared with no drug, as no control is given. As there is no control, so we cannot completely compare the effect of the drug. Alright.